So looking at chapter 3, here we're dealing with cells now. So we've done atoms and molecules and covered some terms and anatomical terms and directions and that sort of thing. So now we're starting to get almost ready to do the actual anatomy and physiology. Not that this isn't part of it, but remember, we have to work our way there to make sure you've got all the basics. And so the next part obviously is cells. And then after this, we'll do tissues, and usually your first organ system is skin. And then you're just going to continue on through the course doing organ systems after organ systems, okay? These are your learning outcomes. Again, take a look at these. This table 3.3, this is referring to the course textbook as per Dr. Tice. If you don't have it, it's okay. You'll need it for AMP2. As far as I know, I'm not teaching AMP2. So make sure eventually you do get that, but this is what this is referring to. For me, of course, pay attention to the study guides and get ready for the tests in that way. But these are what you should be able to go through. So I will work on a study guide and go through these learning outcomes like I've been saying. All right, make sure they make sense to you. Because as you move on to AMP2, that is going to be what is expected, all right? So first of all, getting into cells, the first thing you're going to have to start to do is identify cells by their appearance, all right? So you need to be able to look at these, and you could probably get some examples of each kind, all right? So if you see the term squamous, you'll know that it is a very flat layer of cells, polygonal or polygonal, I don't know how to say that properly, but that just means that there's quite a few sides if you're going to look at them from top down. You could say that squamous cells look like this too, squamous from the side and top down look like that, but there are other situations where you'll have these kinds of cells. So they're kind of oddly shaped. But they usually form a monolayer of some kind. They're all attached to each other. They're not like they're out in the middle of nowhere on their own. Cuboidal are cubes, right? So you can see that there. Columnar, the same thing. Very often they have little appendages on them, which we'll talk about later. Spheroid, they are spheres. Blood cells, very often, yes. Except for red blood cells, which would be classified as discoid because they look like discs, all right? Stellate, a classical stellate type of cell would be a neuronal cell body. Fusiform or spindle-shaped, these would be like smooth muscle cells. And fibrous, that would be like skeletal muscle cells. Or not so much cardiac muscle, but really when you're thinking about really fibrous, very often muscle cells are called muscle fibers. Even the smooth muscle cells are often called muscle fibers, but but this is what you would call fusiform, and this is what you would call fibrous, all right? So you can see they look different. So most cells are about the same size in terms of their diameter, even though they look different, 10 to 15 microns. I'm not gonna expect you to know that, but I want you to appreciate how small that is. And there is an upper limit to the size, and we're gonna talk about that in a sec, why cells are small. And then, of course, the cell shape correlates with function. That comes back to that whole theme in AMP where structure and function are related, all right? So now, the other thing to note, and here's another slide looking at a little bit more detail, all the different types of cells, all right? So here you can see those different shapes and different places where you might find them, right? Now, the thing that I really appreciate, though, is even though cells can come in all of these different forms, when you're talking about human cells, they pretty much all have the same organelles or parts in them, right? Some of them have, extra ones like flagella or cilia or things like that. But when it really comes down to the organelles, they still have the same organelles if you're talking about eukaryotic cell, which would be in human cells. Sometimes they'll lose one of those organelles, like red blood cells is a classic example because the mature red blood cell doesn't have a nucleus for a number of very important reasons, but they had a nucleus when they were developing. So they still have all the organelles, right? So we will talk about all those different organelles in this chapter. Now, first of all, why are the cells small, or what motivates them to stay small? And almost when they get to a certain size, they divide. And not all cells do divide, right? Neurons don't tend to divide, and skeletal muscle cells don't tend to divide. That, you know, these kinds of things. But cells that do divide, one of the motivators for division is the size. So once they get to a certain size, they'll split. Now, why? Now, the thing to think about here is that most things like nutrition going into a cell, gases like oxygen going into a cell, wastes leaving a cell, these all are done with a passive process called diffusion, which we'll talk about in a sec, all right? So in order for this, and it has to be really efficient, for the cell to survive, it has to be efficient. And the way to ensure the efficiency of that is that the cell has to have a large surface area to allow for these things to go in or out, and a small volume that these things need to pass through to get to the cell edge, okay? So they need a large surface area to volume ratio, all right? And this slide here is just to show you how that can work, right? So if you imagine that a cell is a cube, so say it was a cuboidal cell and it had some sort of unit, it's not going to be a centimeter, right? But we'll just say that the unit height, width, and length was 1. You know that if you added the surface area to that, it would be a total of 6, because it's 6 sides on a cube. And the total volume, 1 times 1 times 1, would be 1. So the surface area to volume ratio would be 6 over 1, so that would be 6. Now, say your cell is growing. Now, just say it would just grow to occupy a volume like this 5-unit measurement cube. So a 5 by 5 by 5 cube. Now, if you worked out the surface area of that, the total surface area would be 150 units, whatever they are. And the volume then would be 125. And the surface area to volume ratio now would drop down to 1.2, right? Whereas it was 6 before. So you've not maintained or increased your surface area to volume ratio. However, if you tried to occupy cellular space in the same way, but instead of having one massive cell, but rather filling that same space up with 1 by 1 by 1 cubes instead, your surface area now would go up to 750. Your volume would still be the same, 125. All right, that's what we're showing you here. And the volume, surface area to volume ratio would be maintained at 6. So here you'd say, okay, maybe with this hypothetical cell, the surface area to volume ratio needs to be 6 for it to be efficient. And when it gets to that, it will divide, right, as it tries to get bigger than that. So this would be like an instigator. And it sort of explains to you, how you can take up the same volume, but still maintain that surface area to volume ratio. And hopefully that makes sense to you, especially as we start talking about transport through the cell membrane. So this will make sure that the cell will remain efficient in the transfer of things in and out of the cell. Okay, now we're going to go through the organelles. We're going to spend a lot of time on the cell membrane, or the plasma membrane, same thing, all right? And this is probably unusual, but if you've probably studied the cell before, you're like, oh, well, it's like this, membrane around the cell, and that's all you really talked about. But it really turns out to be all the parts of the cell are important, but the cell membrane is hugely important because this is where things get in and out, and how a cell interacts with its environment, how a cell moves around, is contingent upon what's going on at the cell membrane, all right? Now, the cell membrane, we talked about already when we did our biomolecules, is lipid, right? You know that the main component that forms the cell membrane are the phospholipids, and we saw this before, right? The phospholipid has a hydrophilic head group, which would be the phosphate group, and some other thing attached to it. And then the hydrophobic end, and the 2 fatty acid chains sticking out, that of course is fat, and that would like to dissolve in itself. So like dissolves like, right? So these fatty acid tails associate with each other in an aqueous environment, which you are, which cells exist in, and that leaves the phosphate head group on the outside. And I'm not sure if I said this in the last video, but phospholipids will associate like this spontaneously. Excuse me. And that then forms your cell membrane. But that's not all that's in the cell membrane, all right? So I'm going to get into that in a sec. But now I hope if you look at this, you can appreciate that if something is fat, right? So if it's a steroid or something like cholesterol or whatever, it is okay in this fatty area. So fatty things can get through here really easily, right? They're not easily transported in blood or in an aqueous environment, but they can definitely easily get through cell membrane because the cell membrane is mostly fat. Okay, and I want you to also appreciate that the cell membrane is very fluid. When you see these pictures like this, it makes it look like it's like this wall of fat, like a sliver of butter or something like that, but it's not. It's really, really liquidy. It has about the consistency of salad oil, all right? And in another slide further, I have a link to a video that you can watch where they actually pull out pieces of a bit of the cell membrane and move it. So you can see how fluid it is, all right? So I encourage you to watch that video, but I'll remind you when I see it, okay? So this is really oily, so it's not like lard, okay? So it's more like oil, all right? Now, obviously we need to get things through here. Not everything is lipophilic, so other things have to be existing in the cell membrane to make it complete, okay? So you have this bilayer of phospholipids. That's what forms the structure. But there's two other components in the plasma membrane that you need to remember. The next one is cholesterol. And you'll say, well, Dr. Vora, that's still fat. And yes, it is. But remember, cholesterol has that unusual shape, right? And this picture will remind you, right? It has the 3 6-membered rings and a 5-membered ring. And you see when it comes in here, it sort of sticks itself into these fatty acid tails. And it has a double function that seem opposite, all right? But it works, all right? So this is to imply that there might be some sort of an R group here, okay? Not R group, functional group, all right? So what does cholesterol do? Well, cholesterol restrains the movement of lipids in warm temperatures and in the membrane with many unsaturated fatty acids, right? So you have to say this kind of like structure. And I said to you that this is very liquidy already, right? So if you stick in some cholesterol there, you can see it's going to sort of stabilize out these tails, right? So it will keep it a little bit more solid when it's warm. And you know, oils, when the temperature goes up, they get even more liquidy, right? So you don't want that to happen because then your cell membrane will be too liquidy, right? It needs to be just the right consistency to function properly, all right? The other thing that it does is that in colder temperatures or in membranes that have a lot of saturated fatty acids, it makes sure that the membrane remains fluid enough, okay? So it has this double sort of opposite sort of function, but if you look at the structure, it should make a bit of sense to you, right? So this is why we need cholesterol. So you can't cut cholesterol out of your diet and think you're going to be healthier. You need cholesterol for a lot of reasons, not just to make your steroid hormones, but also to maintain your cell membranes. And where would you be without your cells? So, and no cell membrane, no cell, right? So this is really, really important, all right? So two components, phospholipids and cholesterol. The third one, okay, so remember, you need to be able to get things through this membrane, right? So obviously we need to have a way of moving things that aren't fatty or fat soluble through here. And the way we do that is using proteins, all right? So your third component of the cell membrane are proteins. And like I said, very often these proteins, what they do is form pores or channels to allow things to go through, but they have other functions, which we'll talk about, all right? So here we'll talk about, you know, proteins in the membrane, how to regulate the control of passage of molecules, all right? And some of these proteins, right, they'll go all the way through what we say the transmembrane. Some of them are just membrane, don't go all the way through the membrane, they'll just stay on one side of the membrane, so they're not transmembrane proteins, but they're also still embedded in the lipid, all right? And they have different functions, all right? So suffice to say, you can have proteins that go all the way through, you can have proteins that are on the outside, other proteins that are on the inside of the cell, depending on what the function of that protein is. And like I said, we'll go over those functions. We've got 6 different functions of membrane proteins. All right. So at the end of the day, what you have is this situation where you have cholesterol, protein, and phospholipids making the cell membrane. And it's something that we call a fluid mosaic model, meaning that it's fluid. And remember I told you it's very, very liquidy. So these things are not static. They're moving around, and it's a mosaic, meaning that these proteins are embedded into this membrane. It's not like a sandwich, right? Before they actually knew how this worked, they had all these different ideas of how the membrane might be structured, right? So they could see this is what it looked like by electron microscopy, but it doesn't really tell them where the proteins are and where the fats are and how it's structured, all right? And there's different ways of doing that. If you want to know how they figured it out, I will gladly tell you, but you don't need to know it right now. But if you want to know, I will tell you, okay? I'm not going to tell you right now. Come see me, send me a message, and I'll explain it to you, okay? Now, the other thing that you'll find is sugars, particularly on the outside of the cell. So sugars, they're going to be attached to the proteins and attached to these lipids, depending on what kind of sugar it is and what kind of molecule you're making. So we call them glycoproteins, glyco for sugar and proteins if they're attached to proteins, or glycolipids if the sugar is attached to that. And they have also other functions that we'll talk about in a little bit, okay? So at the end of the day, this is really more what it looks like. And I want you to still imagine this is moving, all right? These things are all these chunks of protein are moving around in the membrane. The lipids are moving. It's not static. It's constantly moving. And there's ways of trying to restrict the movement a little bit, relying on the cytoskeleton. But just imagine this. This is not static, all right? And this is the link to the video, so you can see the consistency of that plasma membrane, so you can give that a look. Now, the other thing I want you to appreciate from this is that the cell membrane really is the communication area. It's not just a boundary. It's a way that the cell interacts with its environment. So out here, you have all kinds of other proteins. Collagen is just one of them. There's hyaluronic acid, fibronectin, all kinds of proteins and sugary proteins out here that make up this extracellular matrix. And we don't even talk about this in this course, all right? But it's so important for cell movement, for cells to understand where they are. This extracellular matrix helps neurons guide their axons during development and helps cancer cells do nasty things that they're not supposed to do, like moving around and stuff like that. So the extracellular matrix tells the cell where it is, okay, and what it should be doing. So it's really, really important. And you can induce breast cancer in mice if you change this extracellular matrix. It's just so crazy. Or you can control breast cancer cells and have them be more normal if you change this extracellular matrix. So it's really, really important. But we don't really even talk about it in this class, but I'm mentioning it to you, all right? Because it's not part of the cell, it's what's outside the cell. Now, I also want you to notice we are going to mention the cytoskeleton, still not very much of that, but here's your cytoskeleton. And notice that the proteins can provide linkages between the inside, the cytoskeleton is, and the extracellular framework out here, the extracellular matrix, which again is hugely important, a huge field of science. And that's what I did for all of my research was involving, how cells interact with the extracellular matrix and what happens on the inside. So I spent many years, 7 years of graduate school, another 5 years postdocing. So you can do the math, right? So, over a decade of time, I spent just studying these interactions, all right? And how the membrane proteins do that, okay, in cancer cells. So it's really important. And if you really want to learn more about it, I'd love to talk to you about it. But this is my one chance to let you know officially as AMP students that there's a lot more going on here than we're even talking about here, right? But the idea of AMP1, and as you go further in your studies, you can gradually layer on your understanding and build that so you become a better health professional. And if you're going to work with patients, you can explain things to them in a better way, all right? But if that, of course, is up to you, how educated you want to be, right? So, or what kind of nurse, or what kind of PA, or what kind of doctor, or what kind of physical therapist you want to be, right? So, but I'm just planting that seed out there. So there's so much more to learn here, okay? Now, Now remember, another job of the cell membrane is to maintain the gradients that we were talking about earlier, right? You have to have gradients across the cell membrane for homeostasis. We're going to learn a little bit about how this works, right? So this is just, I've used this slide before. I do this a lot, right? So this is going to remind you, oh yes, gradients, cell membrane, really important, maintain gradients, all right? You need to keep your homeostasis, you need to keep your balance in order to have a healthy system. Okay, so that's what's reminding you here. Now, let's talk about what kinds of functions these membrane proteins can have, right? We already said some of them are going to be channels, right? They're going to allow things to get through the membrane that wouldn't otherwise be able to get through, and they're going to be open. Sometimes we call those ion channels or pores. And it's aqueous down the middle, they stay open, and as long as the ion is small enough, it can just go right through, right? So it just this makes the membrane kind of leaky. And that's an important thing to remember as we get into how muscles and nerves function later, all right? Then some of them are carriers. So carriers are a little different than the channels or pores because they're more geared to specific types of molecules or atoms and let them go through, all right? And not other ones, right? So sometimes they change shape in order for this. So one will bind on one side, the protein will change shape and deposit it on the other side, all right? So carriers are, and you can see here, sometimes they're also known as transporters, all right? That doesn't make it easier for you knowing that there's different names for the same kind of protein, but that's why we're explaining that to you, all right? Now, receptors, these ones will bind, so we say bind some sort of a ligand. You can imagine, say the ligand is a hormone or a neurotransmitter or something like that. And it binds to one side, and then the protein here does perform some sort of function, and that then carries a message into the cell. And we will talk a little bit about this, okay, and how that works. And this can regulate how the function of the cell, it can regulate the functioning of the cell, right? So here it says, it gives the idea that kidneys can become more permeable, cells in the kidney can become more permeable to water or something like this, or proteins or something like this, right? So there's a regulation here. But receptors are usually a way that the cells communicate with the outside, and then some sort of change on the inside, right? Not always, but that's part of it, okay? Sometimes they're enzymes. You can have enzymatic reactions localized specifically to the cell membrane if you have this as a membrane protein. And indeed, one of the things that I study when I'm talking about, I told you how proteins interact with the environment. One of the proteins that I study is an enzyme. It's a class of enzymes called metalloproteinases, right? So they would be like these enzymes that are localized specifically to the membrane. Okay. Others are linkers, and I've studied different types of that too. So they interact with something on one side and then something else on the inside. So an extracellular matrix protein and a cytoskeletal protein on the inside. Very often important for structure or movement, and how cells bind to each other and interact in different membranes, for example. So there's lots and lots of these. And then identity markers, which is really important for immunity. So specific immunity, how your body knows your cells from somebody else's, right? So there's these proteins here that are involved with that. MHC stands for major histocompatibility, right? So that has to do with immunity. And there's identity markers, all right? So that's another thing. That's a huge field. You could actually study that for a whole semester. So, but you'll get a little taste of that later. Okay. Now, so the plasma membrane can regulate the way things pass through, right? So here in blue, those things pass easily. So water is unique that it can do that because it's polar, right? But there's even special proteins that can be inserted to make the passage of water really quick. They're called aquaporins, so those would be channels. Gases can get through. All right, because gases, remember H2, N2, O2, they're actually nonpolar. Small fat soluble molecules, like I was saying, so like steroid hormones can get through. Then if you have small molecules and they have a charge to them, they're going to have trouble. So that's where you're going to start getting some carriers, right? Pores are going to start doing that. And then large things, large water-soluble things like proteins, they're not going to get through. All right. So the way they do their jobs is by binding to receptors and then causing a change on the inside, right? So we will talk about that, but they cannot get through. All right. Okay, so cells, the proteins that the cell needs, it has to make for itself. So these are the types of carriers that we've got. All right, so we're going to go through these ideas. So we've got, so here's supposedly your lipid bilayer. You can have simple diffusion, and we're going to go over that, but basically here's your concentration gradient, right? So simple diffusion is when substances are going down their concentration gradient. And then we have passive transport. So here you're going to have your proteins involved. So you're going to have channels or carriers. And then things will still go down their concentration gradients, but you can have like a channel or a pore here. Let's go through carrier-mediated, right? So here you can, what did we call them here? Let me go back because different books call it different things. Where was I? Here. Carriers or transporters, right? So that's what I thought. We had carriers or transporters. They're going to change shape. So here you've got this change of shape. So it binds on one side and then we'll release it on the other side. But it's still going down its concentration gradient. So we call this passive because it's not requiring any energy input. Active transport, again, is going to use a carrier or a transporter. But now what's happening is that you're going to pump. Very often these transporters or carriers are called pumps. You're going to push it against its concentration gradient. So you're going to need an energy input. So it's kind of like when you've got a dam and you've got to push the water back up and then it's going to come down again. You've got to put some energy in so that the dam can generate even more energy, right? So this is what we call active transport because it needs ATP, all right? It needs that energy transfer from ATP to allow this to take place. So it's active. And there's several different types, so we're going to talk about that as well. All right, so this is the difference between passive and active. Diffusion. And technically, this is more like dialysis because you've got a cell, you've got a membrane here. So just so you know, yeah. Okay, now simple diffusion. All right, and when I teach 105, this drives students crazy, I know. But simple diffusion is the movement of something from a high concentration to a low concentration, okay? And there's a number of things that will affect that, right? So this is the old, put a drop of dye in here and then watch it diffuse through the water, right? So the more time you have, it will start and it will take up the whole space. So that is simple diffusion, all right? Now, technically, what we have in this case is the addition of a bilayer. And I don't know why it's not introduced in this course, but if you have simple diffusion and you add a membrane to it, you're actually got dialysis, all right? So dialysis, you introduce that membrane, so not everything is going to be able to get through it, right? Because you've got your membrane, it's your cell membrane. So it will sort of regulate what things can go through, and if things are small enough, they'll pass through, right? So technically the cell membrane really is dialysis. Okay, when we're talking about simple diffusion. So simple diffusion plus a membrane is dialysis, all right? Then protein mediated, okay? So here now you're gonna add in, so simple diffusion, I should write down dialysis here because that's really what this is, all right? Then we have channel mediated or carrier mediated, right? So here you've got the addition of the protein, right? So these two together, we call that facilitated diffusion. And I want to see if I actually wrote that term down in here. So protein mediated, also known as facilitated diffusion. You see that on page 3 of my notes, okay? So you have, in this case, fluid-filled pores or channels, or carrier-mediated or gated channels, and that's what you've got here, right? So these are the ones that are going to change shape once they bind to this looks like a sugar, right? Then it binds the sugar and then we'll open up and let it through, all right? Here, this one, the channel or the pore is open all the time, all right? So as long as it's small enough to fit through here, it'll go, all right? So that's the difference here. But all of these are passive. They don't require any energy input, all right? So here we have facilitated diffusion or carrier-mediated transport. These require a cell membrane and protein specific to allow things to go through. Doesn't require ATP. Transport is down the concentration gradient, all right? Now, sometimes, and this is kind of neat, sometimes only one thing will go through, and we call that uniport. Sometimes two things go through, and they're both going in the same direction. We call that symport or co-transport. And then sometimes things will go through, but opposite directions, and that's called antiport or counter-transport, okay? And what's limiting this ability for things to go through is how many of these proteins you have, right? So you can actually saturate that. So saturated meaning that you can actually have them all so busy, and they can only work so fast, right? So depending on how many proteins that you have and how much of the solute that needs to go through, you can actually max them out, all right? So they are limited in how much they can transport by how many of them there are. Hopefully that makes sense to you, okay? Okay. Now, there's this other idea that we have in the body, and we call that filtration, all right? So this is sort of like what I was introducing with the idea of dialysis, right? So you've got diffusion, or movement of molecules, and you've added in the membrane, all right? You've added in cells, all right? So here in this course, we think of it as filtration. So you've got a semi-permeable membrane, right? You're gonna a membrane that's gonna let some things through and not others. And that can be based upon those proteins, right? So different size particles will be able to be filtered than others. And remember, you know, the amount of pressure that's going to be produced here, because this is an aqueous medium, is called the hydrostatic pressure. So this hydrostatic pressure, and as we get further into the course, and especially when you start doing blood pressures in that, you're going to learn that there's a hydrostatic pressure pushing on the blood vessel from the tissue, and another one pushing out from the blood towards the tissue. So hydrostatic pressures are going to affect how well things are going to filter, okay? But what's happening here is that there's no, technically for filtration, it doesn't involve transport proteins, right? So you've got your vessel, your cells, your membranes, and things are going to go through that in response to the pressures that are involved in the blood vessel, all right? So that we call filtration. So some things are going to get through. If they're small enough to get through, they'll get through, all right? And here in this picture, you can see that things are going between the cells in this way, right? And how well things are going to be able to get through that barrier. So here you have your blood vessel, and you've got pressure here, and it's all aqueous, and some things are going to be able to get through and other things not, all right? So that's what we're referring to in terms of filtration, all right? And you've got this semi-permeable membrane that is imparted by the cells, all right? So when we're talking about filtration, we're talking about more along the lines of what's happening in this case, right? Versus what I was saying before with dialysis, which is diffusion across a membrane, right? A semi-permeable membrane, because it's going to let some things through and not others, okay? Depending on the size of the particle, all right? So that would be more like dialysis. And here you can see it's going sort of in between the cells, and it's sort of like, being pushed out, all right? And here we're just showing you the pushing out from the filtration, but later on we'll talk about how it's, there's a pushing back as well, because there's pressure out in the tissues as well, okay? So the size of the particle is going to affect that, how much that pressure is being generated against the liquid medium that these particles are in. It's not using ATP, all right? Things are being forced, right? And there's no transport proteins per se, because you're going sort of leaking through the cells, all right? So hopefully filtration dialysis is a little bit different. And, dialysis is one of your learning outcomes here. But I'm just mentioning it to you so that you understand, okay? Now, of course, that then comes osmosis. And this is a really weird thing, all right? Because usually we say, oh, well, it's the diffusion of water across a semi-permeable membrane, right? Or it's the dialysis of water, right? So the thing is, it's not quite like that, and it gets a little bit confusing. So in the homework, there's a video for you to watch. The professor who talks about this process is sort of explaining to you why it's not really the diffusion of water, because there's more things involved. But saying it's the diffusion of water across a semi-permeable membrane makes it easier for your brain to wrap around what's happening, okay? But I want you to appreciate it's not technically what it is, all right? So let's talk about it. So osmosis, you know, when we're talking about the human body, we're talking about water, okay? So there's going to be questions in the homework that they say, is this osmosis? Is it the solvent, or is it, you know, the solute dissolves in the solvent? I mean, technically, I guess from a physics perspective, I mean, osmosis could involve other solvents, but your solvent is water. So the answer will always be, if there's no water mentioned there, it's not going to be osmosis, okay? So just to help you. But I want you to understand this, right? So water is still going to be going down its concentration gradient, okay? And it's weird to think of it this way, but if you look at the classic YouTube, all right, and then we are going to talk about other aspects of this shortly. Now, say you've got a semi-permeable membrane, and this membrane is permeable to water, but not to what this other stuff is, and it's very often sugar. All right, so something that's too big to get through this membrane. So obviously, if you look at this and you see the same volume, and on this side you've got something else here, technically the concentration of water on the right side here is less than the concentration of water on this side. So that will sort of drive water to come through here. Okay, now if you watch the video, it puts a layer of complexity here, because water isn't just going one direction, it's going both directions, because this membrane is permeable to water, all right? So the video is talking about how when water bumps into this, it's going to create sort of like a physical process where it's going to draw more water in here, sort of like the turbulence factor is going to pull the water in here. So yeah, physics involved, well, yeah, it's true. And it is important to understand that there's the water is going both directions, right? It's the same thing with diffusion, that things are going both directions. But understand that here the concentration of water is still less. So that is a good way to think about it, right? So the water is going to be attracted to this side, and you could think of it as simply due to concentration, but also because of these other particles here. Okay, and what's going to happen then is, as more water molecules come on this side, and they're going to be affected, the volume, by these other things here, the volume in the U-tube is going to go up on this side. Okay, so it looks pretty magic, right? And you know, indeed, back when I did alchemy, I could see some alchemists using this as a magic trick with you don't see me, but I'm doing my bunny ear quote things with my fingers. All right. Now, understand that this movement of water creates a pressure or a force that's pushing. It's good. Remember, water likes to hold on to itself, and it's going up this U-tube, and that exists. That pressure is called a hydrostatic pressure, right? And then you can push down on that, and you can measure what that force is. Like, you can try and push those water molecules back again, because this membrane is permeable to water, so you can push them back, right? And this applied pressure, right, to find out what the force of the water is, the hydrostatic pressure, like what is that? What is the pressure of the water? This is called the osmotic pressure, all right? So the pressure due to water's flow is the osmotic pressure, and it can be measured by applying a counter pressure to see what that is, and how much force is it going to require for me to push those water molecules back over to the other side of the U-tube, okay? So this is the osmotic pressure. And there's osmotic, or sometimes it's called oncotic pressure, on both sides of a blood vessel. And we'll talk about it later. Sometimes it's forcing liquid in, and other times it's forcing liquid out. And osmosis is involved with this, as well as just pressures due to the beating of the heart. So this is some really interesting physics that takes place. All right, you'll see when you take this that in the end, the heart, the pressure, the hydrostatic pressure being pushed into the blood vessel forces more water out than osmosis will bring back in, okay? So then you have your lymphatics to collect that fluid, and if it doesn't work, you get edema, all right? So it's quite complicated, but if you try to just keep it as simple as possible. So just so that you appreciate how this works. So osmosis is always the movement of water, and it happens to be down its concentration gradient, and there's always that semi-permeable membrane involved, right? You don't see a membrane in the description. It's also not osmosis, all right? So you need to have the membrane. It needs to be water, all right? So that should help you. There's some weird thing in the homework where that isn't the rule, and if you get it wrong, let me know, okay? But yeah, okay? All right, oncotic pressure. Now let's talk about tonicity, right? So this is what happens, right? So in an isotonic solution, you have the same amount of salt, or the same amount of solute on each side of the membrane, right? You've got your cell membrane. In this case, they always use red blood cells to show this dramatically. And remember, it's at equilibrium. So there's the same amount of water going in and going out. It's not that the water isn't moving anymore. It absolutely is, okay? And then the cell's happy, right? Human cells need an isotonic solution. Plant cells don't like that so much. But human cells like it isotonic. So same amount of salt, and those don't have to be the same salts, right? So we're going back here. Remember, you do still have your gradients, okay? So that doesn't necessarily mean that the concentration of sodium is the same on both sides, but the salt's concentration is the same, all right? So the things that are going to be causing water to move across the membrane, all right? Ooh, that makes sense to you. That could be all kinds of different ions, all right? Now, if you have a situation where the salt concentration is lower on the outside than it is on the inside, okay? So that means that the concentration of water is lower on the inside of the cell. What's going to happen is that water is going to go into the cell, right? So I always like to say that water follows salt. That's probably the easiest way to remember it. So a hypotonic solution always means low salt. Water follows salt. So where's it going to go? It's going to go into the cell. And that's going to cause it to burst, or what we say, if it's a blood cell, red blood cell, it's called hemolysis, all right? Turns out plants like this because they have cell walls, and then the pressure pushes it back from the cell wall on the cell and stabilizes it. But human cells don't have cell walls. So in this case, when water follows salt, goes into the cell, it causes the cell to burst. Hypertonic, hyper always means more than something. Well, hypertonic solution means you have a high concentration of salt on the outside, right? And a lower concentration of salt on the inside. So now again, the concentration of water is less on the outside, so the water is going to want to go out, all right? And that then will cause the cell to shrink or crenate, all right? So again, water follows salt. That will keep you grounded as long as you know that hypotonic means a low salt solution, and a hypertonic solution is one that is a high salt solution. Where does water go? Well, water follows salt, or it follows solute, okay? So hopefully that will help you. Okay, now let me take a look at my notes. Where are we at here? Okay. Active transport. So remember, active transport, you're using ATP, okay? What you're doing now is that you're pumping something against its concentration gradient, and you're using that energy transfer. You're splitting off that phosphate, making ADP, and in so doing, you're releasing the energy from that high energy bond, and that's going to help drive the substances against their concentration gradient, all right? So very often, here we're saying primary active transport. This is where you're actually splitting the ATP. Very often, these are called pumps. And the one that you really need to learn is the sodium-potassium pump, because it's on every cell in the body, and it's responsible for maintaining this amazing gradient right here, right? Sodium and potassium, all right? Sodium potassium pump maintains this, all right? We've got other ones for this, all right? So sodium potassium pump, all right? Now this is how it works. And let me get here again. All right, so what will happen, and it's a cycle, so what it's doing is it's transporting 3 sodiums, and hopefully by now you recognize this as sodium. It's going to pump 3 sodiums against its concentration gradient from the inside of the cell out, and then it's going to have a different shape. Now it's going to bind 2 potassiums, and it's going to bring those in, all right? Now also against its concentration gradient. So both sodium and potassium are being pumped using the energy of ATP against their concentration gradient, all right? So this is primary active transport, meaning that we're directly using our ATP. So let's look at this in a different way, all right? So you start off with sodium on the, the pump now open to the inside of the cell membrane, and 3 sodiums are going to bind. Now these things have a positive charge, and they're going to have an effect on the shape of this protein. So when they bind, what's going to happen is that the protein is going to change its shape, and that's going to allow for this high energy phosphate to bind as well, right? And this is going to now close, all right? So there's sort of an enzymatic function to this protein, all right? Now, what's going to happen is that it's going to shift with the binding of this phosphate, and it will now open to the outside of the cell. This shape change is going to cause the release of those sodium molecules, all right? And now we can bind our potassium. So 2 of the potassiums are going to bind. And this is then going to cause another change in shape, right? That change of shape is going to cause the phosphate to be released, all right? And then this is going to go back to its original shape, and it's going to deposit the sodiums on the inside of the membrane. I mean, the potassiums on the inside of the membrane, right? So sodium is going to go out, potassium is going to go in, all right? So let's do that one more time, okay? So here you have the sodium-potassium pump being open to the inside of the cell. Sodiums will bind, they're positively charged. It's going to cause a change of shape. It's going to activate the enzyme. It's going to split off that phosphate off of it. So this pump has an enzymatic component to it. And that phosphate is going to bind. So then this will close now, because this is now a big charge and a thing hanging off here that's going to also cause the protein to change shape. Now what's going to happen is that when this phosphate binds, it's going to open up. It's going to allow the sodiums to be deposited on the outside of the cell against their gradient. When the sodiums are free, potassium can now bind, right? These are going to, again, change the shape of the protein. Off will come your phosphates, and it will then open to the inside of the cell, and your potassiums are going to be deposited on the inside of the cell, now against their concentration gradient, all right? So sodium-potassium pump, all right? Or the sodium-potassium transporter, all right? So this is primary active transport because you're using ATP directly. So obviously there's going to be other kinds of active transport because I'm saying primary, okay? Now, this sodium-potassium pump is really important to maintain that gradient, right? So why? Why is that so crucial? So let's look as an example. So say this is the lumen of the kidney, right? Here's the here's the the lining of your ducts here, all right? And here's the ATP sodium-potassium pump using ATP, right? And it's pumping sodium out and potassium in. Okay, now say this is really important because you don't want to lose glucose out to the body, all right? So here you can have a symporter. So this is here called the sodium glucose symporter, and it's going to bring in glucose into the cell, all right? So you've got glucose out here that you don't want to pee out, right? So you shouldn't have glucose in your urine. So you're going to try and bring in this glucose. And the way you're going to do that is allow, capitalizing on your gradient, all right? So let's go back, look at our gradients, right? So remember you have a high, too many slides. You have a high concentration of sodium out of the cell, low inside, so it's going to want to go in, right? So it's opposite to what you had with that primary active transporter, sodium, potassium, ATPase. It's going to come in down its concentration gradient, and that then I won't do this again. That then is going to allow for glucose to come in against its concentration gradient, okay? So I know it sounds weird, all right? So, but glucose is not, this is still, it's capitalizing on the gradient that was established through the sodium-potassium pump, the sodium-potassium ATPase, that's how I learned it, using ATP, splitting that directly to maintain this gradient. Sodium then comes down its concentration gradient, and that allows for glucose to come in through the special transporter, right? The sodium glucose transporter. And this spot here is what we call secondary active transport. It's secondary because you're not using ATP directly, but active because you are bringing something against its concentration gradient. But in order to do that, you had to maintain a gradient for the other substance. And that was done using ATP. So that would be a secondary active transporter, this sodium glucose symporter. And symport means because both sodium and glucose are coming in together in the same direction. Now this glucose then is going to go out of the cell down its concentration gradient. And since glucose is a larger molecule, it needs a protein to do that. So here you've got the GLUT transporter. It's going to allow that glucose into the extracellular fluid on the other side of the cell by facilitated diffusion, all right? Because in order for it to get through, even though it's going down its concentration gradient, since it is hydrophilic, it needs a protein to help it through, all right? So this is secondary active transport, okay? So if you see in my notes, page 4, secondary active transporters do not use ATP directly, but benefits from the ATP-driven primary active transport, okay? There's also tertiary active transporters in the cell too, and that will come up more when when you're looking at the kidney in AMP2, all right? So there's a tertiary active transporter there, okay? Yeah, so imagine now another transporter benefiting from glucose here, and then also now going against its concentration gradient. That would be like a tertiary active transporter, but that's not what's shown here. Okay, and then here, of course, is your sodium-potassium ATPase or your sodium-potassium pump shown here. Okay, so hopefully that makes sense to you, all right? So here, this is how this works. SGLT, sodium glucose linked transporter, all right? So here what's happening is that you've got, you're capitalizing on your primary active transport that maintains this gradient of sodium. Sodium binds to its transporter. Glucose then can bind to its transporter, or its binding site on the transporter. All right, and then this then changes shape. And when it changes shape, it opens to the inside of the cell, and then both of them are released. Okay, so sodium is released to the cytosol where the sodium is low. Release changes the glucose binding site to low affinity, and glucose is then released. All right. So again, the sodium is going to bind to the transporter. It's high affinity binding site. This then cues in and makes this part of the transporter very attractive to glucose. Glucose then binds to it. That then causes the protein to change its shape. It opens to the inside. Now sodium is going to be released because the sodium concentration is low here. And that changes the binding affinity for glucose on this area, and glucose is let go, all right? All right, so hopefully this makes sense, right? So this is like the sodium glucose symporter, right? The actual name for it is the sodium glucose linked transporter. That's the name for it, okay? Hopefully that makes sense to you. Okay, so now we've seen different ways of getting things through. So remember, we've got antiporters that can pump things or move things in opposite directions. And we've got symporters that can move them in the same direction, all right, across the membrane. And all of these very often are driven by some sort of a gradient that has already been established, such as that involving sodium, all right? Okay, so hopefully that makes sense to you. Okay. Now, there's other types of active transport, meaning it needs ATP, but it doesn't involve proteins per se. I mean, it does involve proteins if you look at how this functions, but now this is what we're talking about in terms of vesicles. Vesicles are membrane-bound structures that contain stuff, right? So wherever they form, they'll either contain parts of the cytosol, like the parts of the things that are inside the cell or outside of the cell. So we have endo and exocytosis. And there's a lot of mechanics involved here. So I don't want to say that proteins aren't involved, because the proteins help form these vesicles and move them, all right? But basically what's happening here is an endocytosis. There's an invagination of the cell membrane, and it will bring into the cell anything that is in the fluid out here and can fit into the vesicle will be brought into the cell, okay? And that's called endocytosis. Exocytosis is the opposite. So you've got some sort of substance, whether it's a protein, say a hormone or a neurotransmitter or something, in a vesicle on the inside of the cell. That vesicle then moves to the cell membrane, fuses with it, and releases the contents to the outside of the cell. So that's exocytosis, okay? So when it comes to endocytosis, we've got a few different types, all right? So the first one is phagocytosis. This is like really dramatic, and you can hear there's a couple of videos you can watch to see it in action. When you're talking about human cells, most of the time what we're thinking of for phagocytosis would be, say, macrophage bringing in stuff. And there's a whole process involved here, and some of this is sort of outlined here, and there's people who study this to find out how it works, okay? But basically what you're having are appendages or pseudopods that extend out from the cell. They engulf whatever it is on the outside that needs to be brought into the cell, and in it comes into a phagosome. And then we're going to talk about lysosomes later, but there's digestive enzymes in here that can break that up, and then you can have these like little leftover bits in here, and that's sort of like a residual body in the cell, okay? But phagocytosis is the engulfing or the bringing in of very large things by endocytosis, all right? So it's a special kind. And here you can see sort of the way it works. So say it was an immune cell, right? So you've got your macrophage. It's going to be sensing, looking for things that look different. If it knows that it's foreign, there's some signals that happen, and they bring that in, and then they digest it out. And some of the stuff is then displayed on the surface. So you know, when you're talking about immunity, this is, you know, and Dr. Tice that's her, she's an immunologist, so she talks a lot about this. But basically then the macrophage presents, you know, a piece of that foreign thing that it ate on its cell surface, so that the specific immune response can recognize that, oh, this is what we have. And this is why you get immunized, right? So that your cells, your immune system already knows, recognizes that, and already ready to mount a defense against it, right? So, but it's bound to this MHC. Remember we talked about that before, major histocompatibility proteins. They display that out for the cells to recognize. And there's two different types. There's class 1, class 2. It's very complicated. But basically this is, you know, phagocytosis. It's one of the functions of phagocytosis. Of course, amoebas, you maybe use them in high school to study phagocytosis. They're not immune cells. They don't do this kind of thing. They literally are eating. Okay, that's how they eat. But this would be an application of phagocytosis in the body. Okay. Pinocytosis or pinocytosis, I've heard of it mentioned in different ways. That's called cellular drinking. So here, this is another form of endocytosis, but it's just bringing in small little bits of fluid. And you can see, watch this video, you can see examples of that. And this just brings in, you know, different fluids out here, and then these can also be digested, all right? So it's not bringing in huge things. And then the next one is receptor-mediated endocytosis. So this is when you're not bringing, you can have regular endocytosis, just bringing in stuff, right? But you can also have receptors on these vesicles that are deposited on the cell surface, and they will look for particular things to bring in. So one of them would be low-density lipoprotein, right? So say it was a kidney cell, and it's going to want to recycle this and get rid of this LDL, okay? And it will bind to these receptors, and then an endosome will be formed around that. And there's this protein on the outside of here called clathrin. So this would be the clathrin-coated pit, right? And then it forms this structure, and then the clathrin can be recycled back to the cell surface. So there's and then again, it's going to be degraded, right? So, so it's a very specific type of endocytosis, receptor-mediated. So just so that you know that you can have a very specific response as well, all right? And then these endosomes look different than just a regular endosome from endocytosis, so, right? Because there's the receptors here, okay? And you can actually see the clathrin on the outside. Right, so they're really, really interesting structures. And again, there's people who study this. I haven't. Of course, I know about it because I studied cell membrane, but really, really interesting. You might find this crazy, but when I was an undergraduate in Canada, it's not liberal arts. So when you're going in to be a science student, that's all you take. And I was in the chemistry program. I literally took a course that was called membrane biochemistry. So I spent 4 months just studying membranes. There wasn't a lab involved with it, but it was just studying this for the whole semester. So this gives you an idea. It sounds complicated. It is, and there's a lot to learn, right? So this comes back to what I was saying before, right? Do I expect you to know all that? No, I don't. But I'm letting you see the differences here, because so you can appreciate what's happening. Okay. And again, if you want to learn more, and keep in mind, I took that course probably 1990, I think was my 3rd year course. So a lot has been learned since then, and it was pretty complicated back then that I could have a whole semester course on that, right? So it's, we can level up that knowledge from the intervening, 30 years, all right? So kid you not, all right? So there's a lot to learn here. Okay, so if it feels overwhelming, don't let it be. I don't want to overwhelm you, but I want you to be aware of these things, all right? So what should you know? Again, focus on the study guide, but make sure you understand that this vesicular transport needs ATP. We're going to talk about that, how that works. Proteins are involved, but not in the actual, this part here. And it needs, since it needs ATP, it's still active, all right? This doesn't just happen on its own magically, all right? So here would be another sort of, block type of diagram on receptor-mediated endocytosis. Okay, so you've got a receptor, it binds to something, events in the cell, again, bunny ear quotes with my fingers, and then in comes whatever it is, and then this is then going to be processed inside the cell. So if you could appreciate this level of it, you're golden, all right? So no worries. Now, I had mentioned to you that there are cells, there's proteins that can't get into the cell, but they bind to the cell, they bind to receptors, and then they cause changes on the inside of the cell, which are then going to cause something to happen on the inside of the cell, all right? And this is a very rudimentary introduction to what we call cell signaling, all right? So you've got these different types of molecules that bind to proteins that are on the cell surface, and then they elicit some change in that receptor, and that then it then initiates some sort of communication or change inside the cell. And there can be many different kinds, right? So you can have something bind to a channel here, and that will open it. This is something that would happen in a muscle cell as well, okay? I don't want to get it too complicated, but this could be an ion, or it could be a neurotransmitter is going to open this up. You can have a situation where you have a receptor, and that's then bound to another component of the protein where there's an enzyme here, and it will do something here. So a receptor enzyme situation, this one is called receptor channel. Then you can have receptor G-coupled protein, and in the homework, there's a video for you to watch to appreciate that. There's a whole bunch of G proteins. They use GTP, and then they do different things, okay? Just that you're aware, there's receptors bound to things called G proteins, and then those G proteins will either open an ion channel or initiate some sort of enzymatic activity on the inside of the cell. Again, the video and the homework gives you more than you need to know, but just so that you can imagine it. And then you can have other ones that will bind to something on the outside of the cell, so extracellular matrix protein. Those proteins are called integrins, and they then elicit a change of shape, and that interacts with the cytoskeleton and causes changes there, all right? So there's these different types of membrane receptors and different kinds of initiated changes that can happen in response to that. Okay. Now, let's see, where are we at? I looked at my notes, so just keep going here. Okay, I guess I'm on page, where am I at here? I guess I'm on page 7. I'm on page 7. There we are, slide 37, page 7 of my notes, if you're following along there. Okay, now these vesicles that we were talking about here, so if you bring in vesicles in all these different ways, they can be targeted. Things can be targeted from inside the cell to go out. They can be targeted in the outside of the cell going in, all right? And there's a couple of videos here to watch on this topic. It's really interesting. Again, there's a whole field of study to do this, but I don't want you know, how much do you need to know? Well, again, I'm not expecting you to learn this. This is advanced stuff. This is, you know, one of the reasons this is in here is because I know that Dr. Tice wants you to appreciate this. Again, focus on the study guide. Don't get too lost in the woods, okay? But watch these couple of videos that will help. But what I really want you to get out of this is to understand that these vesicles are not moving around haphazardly. There's a reason they're being brought in. Once they're brought in, they go certain places. Certain things have to go to the cell membrane, right? So you know, you can have, if you have a membrane type of cell, there's going to be a sidedness to the membrane, right? So there's a, you know, the part that's facing a lumen, another part that's facing, say, the blood, and different proteins are going to be, you know, on different sides, for example. So if we go back here and we talk about here, right? So imagine here inside the cell, you're making protein, and you need to have your sodium-potassium ATPase on one side of the membrane, and you need to have your sodium, what do they call it? The sodium glucose link transporter on the apical surface of the membrane. Clearly, you're going to have some sort of vesicle that's going to have to move that protein to one side or to the other, depending on what it is, right? And this would be done via exocytosis, all right? So you'd have a vesicle that's going to be transported here. It's going to deposit that protein on the right side that it needs to be on of the cell, all right? If it's a cell that has a sidedness to it, okay? All right, hopefully that makes sense to you. So this is not, you know, a willy-nilly thing, all right? There is, you know, important messaging that's going on here to direct these vesicles to where they need to go, all right? So you can already sort of see, you know, you've got these different things. You've got an endosome, and then you've got exosomes, and you've got your endoplasmic reticulum, your Golgi, and, you know, things are being moved around in very specific ways, okay? So let's sort of also look at this, all right? So other things can get through around the cells. So if it goes between the cells, we call that paracellular, all right? Very often there's certain proteins between cells that prevent them from being leaky, so you're not going to have filtration taking place here. Those are called tight junctions, and that might preclude that for most things, but some things will still be able to get through between the cells, which we say are called paracellular. Other things are going to have to go through the cells, which we call transcellular, all right? So transcytosis uses endo and exocytosis on opposite sides of the cell, but paracellular things don't need that because you're not going through the cell at all right? So there's all these different things happening here, okay? Now, with that, we have an amazing cellular diversity in our bodies. Okay, so now we're on page 8, and I put some, you know, lists here. There's like 100 trillion cells in the body, and there's 200 different types of cells, say different sizes, shapes, organizations, different rearrangements of those. And remember, you aren't just human cells. Actually, there's more cells that in you that aren't human. So that's referring to your microbiome, or the bacteria and other things that live on your skin and in your gut. There's actually more of those than there are human cells. And I was listening to a scientist once. I think she says you're like 10% human or something like that. I can't remember. It's a really small number. So there's a lot of other things on you and in you that aren't human, but they're needed for your functioning. All right. So I want you to think of that as well. So that was the point of having this image here. So don't forget, you need your commensal or symbiotic bacteria to make sure that you function well. Not all bacteria are bad, right? Some of them you need so that you function, right? Now this brings up this whole idea of cell-cell communication. And there's different kinds. Cells have to communicate between each other, to itself and across the body to help function, all right? And I want you to contemplate how this works. It could be local communication, it could be long distance. And usually when I mention cellular communication to students, the first thing they say, oh, neurotransmitters or something like that. But there's so much more going on, and I'm just going to introduce that to you, all right? And the way your microbiome communicates with you. I mean, there's this thing I have in my notes here on page 8, the gut and the brain. And there's lots of studies out there. You can look it up. I can help you if you're interested, showing that your gut bacteria can affect your mental health, right? So another really, really interesting thing to think about there. Okay. So how does this work? You can have cell contact dependent signals, so that's very local, right? And that local can be, intracellular or unicellular, right? Or cell can stimulate itself, or it can be paracellular, very close by. But here you got a contact dependent signal, all right? So it requires interaction between two membrane molecules on two cells to take place. You can also have the formation of gap junctions where cells can actually transfer things directly between their cytoplasms. Really important when we're talking about how some cancer cells function, how some membranes function, how cardiac muscle functions, all right? So gap junctions, really, really important. If you can't remember what those are, this is again assuming that you know some of this stuff, but if you want to know more exactly what a gap junction is, again, obviously I can talk to you about that. Yeah, I spent a long time studying that. And also even as an undergraduate, like I said, I had a whole semester course just on stuff like this. And I loved that course. It was really interesting. And then we have non-contact dependent signaling. So here you have paracrine. So like this is what I was mentioning before, where it's very close by. You can also call this synaptic. It's not always synaptic, but if you're talking about a synapse, then synaptic transmission is a type of paracrine signaling. Here we have the contact. Autocrine signaling is where the cell is stimulating itself. And then of course endocrine, which is a subject of next semester. But endocrine signaling is long distance communication, right? So a hormone or a substance is produced in one part of the body, then travels through the bloodstream to someplace completely different. I mean, the whole body is bathed in that signal, but only the cells that have receptors for that will then have a response to that. So that's endocrine, all right? The other thing that you'll see in my notes on page 10, that the signals could be electrical, right? So you have an electrical potential, like what you would have in muscle and neurons, or it could be chemical, right? Like what you would have with hormones or neurotransmitters, right? So that's an important thing to wrap your head around, okay? Now, this is sort of an introductory slide. I'm gonna sort of explain this a little bit better, but you can have, for this signal transduction, we call this signal transduction. This is a whole field. Signal transduction is another field of cellular studies, where you have an extracellular molecule, same hormone, it binds to its receptor. And then as we said back here, those receptors are typically gonna be G proteins of some kind, all right? And then they're going to do something on the inside of the cell to elicit a response, okay? And then one of the responses would be to then produce a molecule that's going to produce an effect inside the cell. So even though the protein or the molecule here, we call that the first messenger, can't get through, it causes a change that produces other molecules on the inside, and those are called the second messengers. So we say these are second messenger systems, okay? So sometimes what these do is then activate a chain of phosphorylations, protein phosphorylations elicited by different enzymes. And when you're phosphorylating a protein, the enzymes that do the phosphorylating, they're called kinases, right? And there's like a whole cascade of these that take place, that are involved on the inside of the cell. and they amplify, because an enzyme doesn't just act on one substrate, it acts on many, right? Remember, it's a catalyst, so it can you recycle it. So this is going to augment the signal, all right? So I prefer to compare this using this slide, so you can sort of wrap your head around it. So say we're talking about hormones, all right? And I already said to you, steroid hormones, right? So testosterone and progesterone are fats, so they can just go right through the cell membrane. And as such, so say you've got, say just pretend this was estrogen, right? Since I'm female, we'll do estrogen, all right? So say, this will actually diffuse through the membrane, and it can bind to a receptor. It can even diffuse through the nuclear membrane, bind to a receptor on this side, and then cause the activation of a gene and then synthesis of a protein. Okay, so you don't even need very much of this. You just need a few of these molecules getting in here. The important thing to understand here is that you're not going to augment what's happening. You're limited here by how many of those steroid hormones you have to cause this to take place, all right? But the steroid hormones have the advantage that they can actually get through the cell membrane. Now, just say you have a non-steroid hormone, right? So that would be called the first messenger, because it's going to bind to the cell. Very often, non-steroid hormones are proteins. They're not always proteins, but very often they are, most of the time. So they're going to bind to a receptor, and then for that hormone to cause a change, it's going to need one of these receptor-mediated changes, right? So this was the first one that was discovered here. This is a G protein. G protein is going to is an enzyme. There's an enzymatic component that is going to take a molecule of ATP, and it's going to change it into something called cyclic AMP. And this is your second messenger. So the first messenger is your hormone. Cyclic AMP is your second messenger. And that's going to, and you're not going to just make one cyclic AMP. You're going to make a whole whack of them, all right? And that cyclic AMP is then going to activate another enzyme, and another one, and another one, and another one. And I want you to appreciate that these are all going to be an increased, an enzyme isn't just going to act on one molecule. It's going to act on many of them. So while this hormone can't get through the cell membrane, it has the advantage that you need very little to cause a huge response in the cell. And you can have different enzymes activated. So you can have multiple different effects taking place inside the cell, all right? And this is what we call a cascade, a signaling cascade, all right? And again, you will watch the video to show you the different types of G proteins, the different kinds of things that can happen. This one here, in terms of cyclic AMP, is the one we mentioned in class because it was the first signaling pathway that was sort of identified this way. And cyclic AMP is what you use for adrenaline, right? So, so for your fight or flight response, this is the one that was identified here. Okay, and these enzymes, very often what they do is they phosphorylate another protein, and these enzymes would be called kinases because that's what they do. So a phosphate can activate or inactivate another effect. Okay, so it's really quite complicated. And I used to joke that people who study that kind of thing, like cellular signaling, they're never going to be done, and every signaling pathway interacts with every other signaling pathway. So this was not something I enjoyed, so I used to try to avoid it like the plague because I thought it was so complex. And I always thought it's a job that was never done. But suffice to say, obviously, there are people who specialize in this very topic, okay? Now, the last thing in this chapter are the parts of the cell. And I'm going to go over this pretty quick. You've probably seen a lot of these before. And if not, you can use my notes. And if you have them for 1 or 5, you've got an advantage because this is notes. But there's only one way I like to teach this. So here, this slide is your good review. Make sure you know what each of these organelles or structures do, all right? I like to start at the nucleus and work my way out. So the nucleus itself, it's not a single membrane system, it's a double membrane system. So what do I mean? You've got 2 phospholipid bilayers there. And your nucleus holds your genetic material. Obviously, that's where your chromosomes are. The chromosomes have genes, and the genes code for protein, right? If you can't remember how this works, transcription and translation, I don't talk about it in this chapter. Very often an A&P course would also talk about this in chapter 3. If you want to review it with me, we'll go over it. Come see me, we'll do that. It's just something that you should know at this point, that that's what happens. But if you really want to know the mechanics of it, if you didn't take 105 with me, come see me if you want to review it. But don't worry if it's not on the study guide. I'm not going to put it on the test, okay? But remember, your genes in your chromosomes code for protein, and you make proteins in the cytoplasm, which we're going to talk about. So you have to get that message or the code out, and that's where you need RNA. You've got ribosomal RNA and messenger RNA and transfer RNA that's going to be required here, okay? Now, in the nucleus, you have the nucleolus, all right? So this is why I'm mentioning the ribosomal RNA. Well, ribosomal RNA is constructed in the nucleolus, which is in the nucleus, all right? So clearly that ribosomal RNA, which makes ribosomes, which we'll get to, has to get out. So how does that happen? And how does the messenger RNA get out? And how do things get into the nucleus when you've got this really thick double bilayer? Well, you've got nuclear pores, okay? So the nuclear pores are these structures that help things go in and out of the nucleus, all right? And here's a close-up view of them, right? Here's ribosomes, close-up view of the nuclear envelope. The DNA in here is coiled into chromatin. If you want a tutorial on the different forms of DNA, I can do that too. So chromatin, chromatids, chromosomes, I can easily do that for you. But again, it's soon you know it, but it's not as critical as all the other things I'm going over with. So just make sure if you really want to know, we'll make sure reach out and we'll go over it, okay? Now, the pores are going to regulate what goes in and out of the nucleus. The other thing here is notice how the nucleus always has this nice shape. Well, that's not by accident, and it's not because it's full of DNA. There's actually a skeleton inside of the nucleus, which is called the nuclear lamina. And this is what it looks like. And it is responsible for maintaining the shape of the nucleus and keeping it nice and open so it can hold everything that needs to be held there, all right? Okay. Now, what happens to those ribosomes? Well, the ribosomes, they are responsible for protein synthesis, right? So ribosomes, they're not just RNA, they're actually made of RNA and protein, but they are responsible for protein synthesis. There's two types, there's free and bound. So free ribosomes exist in the cytosol, or the cytoplasm of the cell, not bound to anything. They do make proteins, and those proteins that they make are going to be found in the cytoplasm. So they're soluble, right? So they're going to be out, and they're not going to be, held within some sort of vesicle, all right? So these are soluble proteins that are in the cytoplasm. But other proteins are membrane proteins, and they are actually made on bound ribosomes, and those bound ribosomes are bound to the next structure, which is called the endoplasmic reticulum, okay? So the endoplasmic reticulum that is holding onto the ribosomes is called the rough ER. So let's look here, right? So now look at rough ER is endoplasmic reticulum. And that rough ER, so endoplasmic reticulum plus ribosomes, happens to be closest to the nucleus, okay? And then you're going to have smooth ER. So smooth ER is further out from the nucleus. Obviously going to be some, so it's not night and day. There's going to be a transitional ER. But smooth endoplasmic reticulum does not have ribosomes bound to it, so it's not responsible for protein synthesis, right? So rough ER, since it's got ribosomes there, protein synthesis occurs there for membrane-bound proteins. But smooth ER, there's no ribosomes there. So what happens there? Well, there we've got several things. Smooth ER is often used to synthesize macromolecules other than protein. So for example, lipids. It's also used for metabolism, detoxification, and it can store things. So the smooth ER in muscle, for example, stores calcium and actually has a special name, which we'll talk about later, okay? So smooth and rough ER. So make sure you know the difference. Now, if you're making a membrane-bound protein in your rough ER, so that is then going to have to be shipped and processed in different ways. So remember I said to you, say we had that cell that had a sidedness to it, a top and a bottom, and things have to be moved to different parts and different things happen to it. That gets processed in the Golgi apparatus, right? So the Golgi is a set of membrane-bound, membranous structures. There's a sidedness to it. So the cis face is the receiving side. So that's the side that's closest to the nucleus. And the trans face is the side that's closest to the cell membrane. And proteins are processed here, all right? There's different things that happen to proteins. So say sugars are added, different things are done. And there's like a shipping system here with vesicles moving from one side of the Golgi to the next, and then off into the cytoplasm to be shipped to different places, all right, wherever those proteins are needed, okay? And that would then bring me to the next thing, are the vesicles, right? So we're here down on the bottom of page 13. So vesicles are stored for use for storage, movement, so endo and exocytosis, processing, so some of these vesicles are going to be called peroxisomes, so they're involved in detoxification, lysosomes, which we're going to talk about right now. So some of them are peroxisomes. Peroxisomes are involved, like I said here, in detox. Lysosomes are involved in breaking down stuff, so digestive enzymes are found there. We also have inclusion bodies, which is sort of like leftover bits that are sitting in the cell. Then, of course, the cytoskeleton, and again, like I alluded to, there's a lot to learn about the cytoskeleton. But what do you need to know? Well, you need to know that the cytoskeleton basically is that network of fibers on inside the cell. Again, it's not static. The cytoskeleton is always changing. But in general, what does it do? Well, it provides a network through the cytoplasm so that things can be moved. It's required for support and movement. And organizes structures. And there's three types of molecules that are form the cytoskeleton. So microtubules, which are made of a protein called tubulin. Microfilaments, which are made up of a protein called actin. And then there's also intermediate filaments. It's not listed here. But intermediate filaments, there's a whole variety of them, and they're different ones depending on different types of cells. So keratin would be an intermediate filament that's found in skin. Okay. Now, how do we move these vesicles around? I said that the vesicles are very specific, and they're taken to different places. The proteins that are being moved by them actually have little codes on them, like addresses, so that the vesicle knows where it has to go. Like, does it have to go to a lysosome? Does it have to go to phagosome? Does it have to go to the cell membrane? All right. And these vesicles are moved using these proteins called motor proteins, as you can see here. And these motor proteins are fascinating. They actually walk along the microtubules, all right? So I know you probably think microtubules are important for cell division. Yes, they are, but they're also really important for vesicles to be carried along and moved to different parts of the cell. And these things, literally, the protein walks along the cable and it carries the vesicle along with it. And for it to do that, it uses the energy of ATP, right? So it uses that energy transfer from ATP to allow for the vesicles to move along, okay? Which is fascinating, how that works, all right? So in your cells right now, there are vesicles being walked around using these motor proteins, okay? There's two different types that I'm aware of anyway. So kinesin and dynein. One goes one way, one goes another way. One goes to the cell surface, one goes towards the nucleus. So very, very interesting how that works, okay? Then we have centroids. You've got two centroids that are always, always, always at right angles to each other in a structure called the centrosome, all right? And if that doesn't fascinate you right from the beginning, like, why is it that they're always at right angles to each other? How does that happen? It's crazy, but it is so, okay? And the microtubules grow out from this structure. And obviously, when you have cell division, the centrosome doubles, and one goes to one side of the nucleus and one goes to the other side of the nucleus. How does that happen? I don't know. But again, something really, really interesting to say. I'm sure there's somebody studying that. I'm not aware of how that works, but I just find it fascinating. Now, what do these things look like? Well, they're also made up of tubulin, all right? And they're arranged, there's like these cables of tubulin that are found here. And you can look at them, there's like, I think there's 9 sets of 3 in each one. Let's count this. 1, 2, 3, 4. I can't see this properly. Do it up here. 1, 2, 3, 4, 5, 6, 7, 8, 9. Yeah. So 9 sets of 3 and tubulin, and they're held there. And how I don't know. But the microtubules grow out from that, right? And they always have that structure. And that's important. The 9 triplets, all right. Now, and the reason I'm bringing that up is because we're going to talk about cilia and flagella right now, but there is tubulin in the middle of the flagellum and in the cilia, all right? So a flagellum is, like you see in sperm, that tail, right? So it helps the cell move about, and the only example of a flagellum in the human system, of course, is sperm. We also have cilia. Bacteria are really good with cilia. They use them for motility. In the human body, cilia are usually found in the respiratory system, and they help move things like junk out of your respiratory system that doesn't belong there. But it's also an extension. It has tubulin down the middle. It's a motile structure, and it has a very different type of movement than, say, a flagella does. But again, it beats in a certain thing. Here it says the direction of organisms' movement, so it was a bacterium. But in the human body, the cilia are going to be beating to move things along the surface, okay? Such as the lining of the respiratory tract. And these things are cellular appendages, the flagella and cilia, that have tubulin inside, okay? So how does that work? So here, say we're looking here at the bottom of one of these structures here. And remember the centroids were 9 triplets. Now in the middle of this, this appendage that's coming off here, I don't know if this is a cilia or if we don't, but anyways, you have up here, you have 9 doublets, and in the middle you have 2, right? So then you've got this, these, and then there's these radial spokes that extend out from that, okay? And that's what's found in here, but it doesn't explain how it moves, all right? So the movement it's pretty wild. So what you've got to have down one set of the doublets is, again, those little walking proteins, the dynin, right? And they walk along each other. They're fixed on one end, and the other side they walk along it. And when they do that, it bends, right? It's really crazy. This requires ATP. Now, to anchor this in place, we have this thing called a basal bodies. You're supposed to know what that is. This is why I have to get into this. So at the very bottom of this, you don't have your central 2 microtubules and your 9 doublets, but you again have your 9 triplets, okay? And you've got spokes that radiate out from that. So that's your basal body. It's found at the base of a cilia and the base of the flagella, and it anchors it in place so that the membrane doesn't get all wishy-washy when these things are moving, right? So you need to keep that stability there. And this thing is going to be anchored using the cytoskeleton so that it can function, all right? So here's an example of how it moves, right? So I said to you that the dynin is going to walk along its neighboring set of cables. And if you didn't have the basal body, these things would just rub along each other. But you got the basal body there, it holds it in place, and then it can allow it to bend, okay? And a flagellum and a cilia move in different ways, all right? Just so you can get an appreciation of that, okay? But yeah, it's really wild, and yes, it happens in your cells right now, okay? Now here's my token extracellular matrix slide. And as I mentioned, extracellular matrix is super, super important. It's so important in terms of the cell understanding where it is and what it is, all right? 2 examples of extracellular matrix proteins are collagen and fibronectin. You've probably heard of that before. I'm also seeing sometimes in advertisements for skin products now, hyaluronic acid. That's another one, okay? And these are proteins that are covered with sugars, and they've got a protein core, sugars on the outside, all right? Really, really important. And what do we use them for? Well, support, adhesion, movement, and regulation, which sounds very trite once you really understand what they actually do. But at least know that, okay? Now, I said to you that proteins and lipids in the cell membrane often have sugars on them. And that sugar coating is called the glycocalyx. Sometimes it's called a fuzzy coat, because if you look at it by EM, it makes the cell look fuzzy, all right? And this is called the glycocalyx. You don't need to know any more than that, but here you can see different varieties of these membrane proteins and these different types of sugar coats and some of the different functions that they can have, right? So they're really important for different communication aspects and for movement, all right? These here are the types of sugars that you can find on them, all right? So, you know, anyways, there's a lot. I don't know. This is galactose, for example. Anyways, there's a lot of different types of sugars and things that are on there, all right? Again, this is another part of, you know, it is in my field and it's not, right? Clearly, there's so much to know. I'm not really good at glycoproteins, but I do know that they're there and they're really important, and they form the glycocalyx. And when I was a student, I used to be able to know which proteins or which enzymes formed which ones of these, but I can't remember anymore. But anyhow, it's there. If you want to know, I can reach into my memory and we'll talk about it, okay? The other thing that is on the learning outcomes are pseudopodia. You don't really see them very often in human cells, but you do see them in macrophage, all right? So this is coming back to the idea of phagocytosis. So when you have phagocytosis and the cell is extending around something really, really big, those extensions are called pseudopods, all right? So really that's just the name of it. They're also used for motility. And that is my field, cellular motility. So you've got a pseudopod that will extend out in the cell, the direction that the cell is going to want to go. And there's a lot of proteins involved with that. So these pseudopods are involved in movement. I was usually looking at that movement in terms of what cancer cells do when they're moving around the body. Yeah, but pseudopodia, that's what these kind of structures are. Usually students see them in amoeba, all right? But I want you to understand that there are cells in the human body that also form pseudopods. And the last structure, I think this is the last structure, is the mitochondrion. The mitochondrion, really, really important. Things to know. Mitochondria have their own DNA, all right? They make their own proteins, all right? So they also have their own ribosomes, all right? Evolutionarily, it was thought that mitochondria used to be bacteria that our cells engulfed because they were useful, because they make ATP, right? So the key function of mitochondria, making ATP. But I want you to understand that these guys divide on their own, got their own DNA, they got their own protein-making machinery. They don't need the cell to do that. So they do their own business. In lab, you're going to have to know the structure of these guys. So understand that there's an inner and outer membrane, all right? So both of them are phospholipid bilayers. The inner membrane has invaginations in there, all right? And these invaginations are called cristae. This is where you have your oxidative phosphorylation to make ATP. So right away, what's happened here is that these cristae also increase the membrane area available to make ATP. So that's why these are really important. The fluid in here is called the matrix. And I'm trying to think. You're just supposed to know that there's mitochondrial DNA, and you know, as opposed to your nuclear DNA, this is the cell. Right? So here's your mitochondria. You can, if you stain the cell for DNA, you'll see these spots out here, and that's what will mark all of the mitochondria inside of the cell. Trying to think of anything else that you need to know for your picture, but you'll see it in the home. If you're in lab with me, I actually have you draw it. But this is what I'm kind of expecting if you're in my lab. You draw something like this and appreciate where all of these different structures are. Okay, and what do? Their job is to make ATP. So here's your final sort of list. I love this video. I recommend watching it. It's called The Inner Life of the Cell. I love it. In that you can sort of see vesicles being carried around on the microtubules with the motor proteins, the dynin. It's fun to watch. Hopefully you have time to watch that and enjoy that. I never get tired of watching that, but That's me. But hopefully you'll look at it once and then you can see how weird I am. But it's really well done, and you can see all kinds of different types of things that I've mentioned and others that I haven't mentioned. So you can see that there's even more to what's going on in the cell, right? So when you're looking at the cell, you've got the cytosol, which is the fluid that holds all of this stuff in there, all right? It's not just nothing. There's stuff there. And then you have the organelles, the cytoskeleton, and the inclusions, all right? And hopefully that does it for you, all right? Again, focus on the study guide. Like usual, there's tons and tons of stuff, but this is A&P. I'm not making this up. It doesn't matter where you take A&P, there's lots of stuff to know, okay? And that's what makes this course hard, all right? Don't ever panic. When you feel like there's too much, come see me, focus on the study guide. The study guide will ground you. The study guide is what's on the test, okay? Other than that, thanks a lot. And just let me know if you need help.